Hello students, I hope you did your mock test well. Myself Nitya Priya, biology faculty from Venper Academy. So now we'll start with the answer discussion. So first question, which one of the following aspect is an exclusive characteristic of living things? So when we talk about this question, so this is very easy question, right? So out of this, option C will be the right one. So when we talk about the exclusive characteristic feature we observe in living things will be perception of events which is happening in the environment and their memory. 102, which of the following is the first step in taxonomy? So when we talk about this question, the first step in taxonomy will be the identification of the organisms. So when we talk about the taxonomy, we know it is the naming of the organisms, right? So where we describe the organisms and we'll classify the organisms. So the first step in the taxonomy will be the identification of the organisms. We need to know what the organism is exactly, its name and the place of the organism. So the first step will be the identification of the organism. 103, in which group of organisms the cell wall forms two thin overlapping shells which fit together? So when we talk about this question, the right answer is chrysophytes. The right answer will be chrysophytes. So when we talk about the chrysophytes, especially we know it includes diatoms and desmids, right? We call them as golden algae. So it will be exactly like two overlapping shells, they are exactly fit together. So like a soap box, you can see that especially in the chrysophytes. Okay, 104, T.O. Diner discovered, so he discovered the free infectious RNA, right? So he in the year 1971 he discovered the viriod, right? So when we talk about the viriod, especially they'll have RNA as the genetic material and they won't have any protein code, and that RNA is of low molecular weight. So for 104 it is option D, which is the right answer. For 105, which one of the following is wrong about Kara? When we talk about the Kara, especially you can see the upper part of the Kara has the female reproductive part, right? Oogonium will be there. And when we talk about the lower regions, you can see the antheridium will be there. So when we talk about this, especially you are just going to pick the statement which is wrong. So upper antheridium and lower oogonium. So as I mentioned you previously, you can see upper oogonium will be there and lower antheridium will be there. So here where that C statement is wrong. 106, male and female gametophytes are independent and free living in. Male and female gametophytes are independent and free living in. So where you can observe, so the options are mustard, castor, pinus and sphagnum. So when we talk about the male and female gametophytes, in this option, you can prefer sphagnum, right? So sphagnum will be the right option. So you can see when we talk about the sphagnum, we'll see that in the bryophyte, right? So the dominant phase will be gametophyte, which is independent. So when we talk about the remaining options, mustard, pinus or castor especially, you can see the main plant body will be sporophyte. But the gametophyte is actually dependent on the sporophyte. So option D will be the right one. So male and female gametophytes are independent and like they are free living in the sphagnum. In case of mustard, castor, pinus, you can see male and female gametophytes are actually not independent, like they are dependent on the sporophyte. 107. If the diploid number of a flowering plant is 36, what would be the chromosome number in its endosperm? So very easy question, right? So when we talk about the diploid number, 2n is equal to 36. Now the question here is endosperm. So the clue they kept you here is flowering plant. So when we talk about the flowering plant, when we talk about the endosperm especially, it is triploid, 3n, right? So n is equal to 18. So 18 into 3, you will get 54. Okay. Then 108, which of the following is not a stem modification? So when we talk about this question, you can see tendrils of cucumber, flattened structures of opentia, thorns of citrus. All of these are actually a stem modification. The one which is not, which won't comes under the stem modification will be the pitcher plant. Right, so when we talk about the pitcher plant, this is actually a leaf modification. So lamina, it is getting modified into a pitcher, so especially to catch the insects. So this is not a stem modification. It comes under there in the leaf modification. 109. In bougainvillea, thorns are the modifications of. So when we talk about the bougainvillea, the thorns are the modifications of stem. So like when we talk about the thorns, like we know like they are hard, pointed, straight structure, like which is meant for production. So axillary bud gets modified to a thorn. So stem will be the exact right answer. 110. The ovary is half inferior in flowers of peach. So like when we talk about the ovary, it will be half inferior, especially in the perigynous, right? So examples will be peach, plum, and rose. So here in this option, peach will be the exact right option. And 111. So like in this question, like we are supposed to match the presentation along with the examples, right? So first, like you can see marginal presentation. We'll have a look on the, okay, here we go. So for marginal presentation, you can see here, this will come, right? Pea plant will come, A5, right? Then like when we talk about the axile presentation, especially, you can see it is observed especially in the tomato and lemon, right? So axile presentation, it is observed in tomato and lemon. So here we go, we got the answer, right? So for so marginal presentation, pea plant will be the example, and for axile presentation, tomato, lemon will fall into that. And when we talk about the parietal presentation, you can see cucurbita will come, right? Mustard will also come there. And when we talk about the free central presentation, so dianthus will come, dianthus primrose will come. And when we talk about the basal presentation, you can see sunflower and marigold will come there, right? Then 112, which of the following statement are correct? So like in this question, like you can see 4 statements are there, like we are supposed to select the statement which are right. So like when we talk about the first option, first statement, many plants belonging to the family Fabaceae are good ornamentals, source of medicines and vegetables. Okay, the plumule and radicle are enclosed in sheath which are called Coleorhiza and coleoptile respectively, and flowers having either stamens or carpels is unisexual. Basal alternate linear extupulate with parallel venation types of leaves is found in the family Liliaceae. So these are the four statements. So when we talk about the statement, especially the first statement is wrong, right? So they belongs to the family Liliaceae, not to Fabaceae. And when we talk about the plumule and radicle, like yes, they are actually enclosed in the sheet, but here it is given as, in case of plumule, it is given as coleorhiza, no, it is coleoptile, and even this statement is also wrong. So the right statement will be third and fourth. Okay, a flower which is having either stamen or carpel is unisexual. Yes, and you can see basal alternate linear extupulate with parallel venation, like it belongs to the family Liliaceae. 113, interfascicular cambium develops from the cells of. So when we talk about the interfascicular cambium, like we dealt with this especially in the dicot stems, right? Especially in the vascular cambium, like we had a look on those interfascicular cambium. So you can see it is the medullary race. Okay, so the cells of medullary race, like they become meristematic, right? They become meristematic. So where they are responsible for the cambial ring formation. So it is from the medullary race. Then 114, during secondary growth of plants, stem phylogen cuts off cells on both sides. The outer cell get differentiated into. So we know this is also from same dicot stem, secondary growth. So when we talk about the phylogen, especially like this cuts off on both sides, the outer cell, like it gets differentiated to cork. Okay, outer side it gets differentiated to cork, or we call it as phellum. And when we talk about the inner cells, like they'll get differentiated into secondary cortex, or like we call them as phellodome. Okay, 115, read the different components from 1 to 4 in the list given below and tell the correct order of the components with reference to their arrangement from outside to inside. So this is also from secondary growth, as you can see here, from outer to inner. So the inner region will be wood, right? We know it is secondary xylem, so the inner region will be wood there. And when we talk about the outer region, it will be cork, right? So phellum will be the outer region, and after phellum, like you can see, secondary cortex will be there, followed by secondary phloem, and the innermost layer will be wood. Okay, this is the order of arrangement from outside to inside. 116, which of the following is not membrane bound organelle? So when we talk about this question, like you can see, ribosomes will be the exact right answer, right? So they are membraneless organelle, like they are not surrounded by any membrane. But like when we talk about lysosomes, mesosomes and vacuoles, like they are actually membrane bound organelles. 117, so a common characteristic feature of plant sieve tube cells and most of mammalian erythrocytes. So when we talk about the common characteristic feature of mammalian erythrocyte, so we know erythrocyte is devoid of cell organelles and the nucleus. And when we talk about the plant sieve tube cells also, like they are not having nucleus. So absence of nucleus will be the common thing between the mammalian erythrocyte and also in the sieve tube cells, right? Then next question will be 118, essential and non-essential amino acid is. So when we talk about this, the essential amino acid is valine and the non-essential amino acid will be tyrosine. 119, so they have given you the image of adenylate acid, right? You can see, right, deoxyribose sugar is there, and nitrogenous base is there, and phosphate group is also there. So in this image, like we are supposed to identify the bond which is present between the phosphate group and to the deoxyribose sugar. So the bond is ester bond, right? So the bond which is present between the sugar and to the phosphate is ester bond, phosphoester bond. 120, with reference to enzymes, turnover number means, like when we talk about the turnover numbers, like we can see, so this is the number of substrate molecules that a molecule of enzyme converts it into products per minute. So in a minute, so within a minute, so how many substrate molecules are getting converted into a product? So that is what we call it as turnover number. 121. During which phase of cell cycle amount of DNA in a cell remains at 4C level if the initial amount is denoted as 2C. So initial amount is denoted as 2C. So DNA synthesis will take place in the S phase. It will be 4C there. And in the G2 phase, you can see the amount of DNA will be same. Only in the G2 phase, like you can see the amount of DNA will be at 4C. Yes, S phase and in the G2 phase. 122, cell plate grows from. So when we talk about the cell plate formation, you can observe that especially in the plants, right? It will be in a centrifugal way. So from the center region to the lateral walls, like you can see, there will be a cell plate formation. 123, two cells A and B, like they have given you the osmotic potential and the pressure potential. Okay, so what will be the direction of water flow? So like when we talk about this question, especially, like we know water potential is equal to solute potential plus pressure potential, right? So when we go with the cell A, the water potential, like we'll get there will be minus 10 bar. And when we talk about the cell B, the water potential, like we'll get there will be minus 12. Right. So when we talk about the water, especially, it moves from the higher water potential to the lower water potential. So you can see the higher water potential will be minus 10 bar. So from minus 10 bar, like the water, it moves to the cell B. So from cell A to cell B. Then 124, loading of phloem sets up a dash that facilitates mass movement in phloem. So when we talk about the loading of phloem, especially, so where you can see the sucrose, like it gets actively transported into the sieve tube. So when the phloem is getting loaded, you can see there will be a water potential gradient, right? So loading of phloem sets up a water potential gradient, so which is responsible for facilitating the mass movement you can see in phloem. 125, the stable compound of nitrogen fixation carried out by nitrogenase is. So like this comes under there in the biological nitrogen fixation, right? So when we talk about the nitrogenase enzyme, this is responsible for catalyzing the conversion of atmospheric nitrogen to ammonia. So the obviously the stable product will be ammonia, right? Next question. Bacteroids are. So when we talk about the bacteroids especially, so you can see them in the biological nitrogen fixation. So they are nitrogen fixing bacteria. You can see they'll be present inside the root nodules. So when a bacteria is infecting the root region, like you can see there will be a curl on the roots where the bacterial cells, like they'll enter into the cortex region of the plant. So there you can see there will be a nodule formation, right? So when we talk about the bacterioids, like they are nitrogen fixing bacteria, you can see them inside the root nodules. 127. Protons are released from splitting of water during light reaction. The number of protons released from splitting of 5 water molecules. Like when we talk about the water splitting process, especially you can see from 1 water molecules, like we'll get 2 protons, right? So if the water molecule is 5 there, so 5 into 2, we'll get 10. So 5 molecules of water, it produces 10 protons. 128, a student added DCMU to an illuminated suspension of an alga and found no evolution of oxygen. Then he added ferricyanate to the suspension and oxygen was evolved. Select the statement that gives the reason for evolution of oxygen. So very easy one, right? Like we know the aerobic respiration and the or aerobic photosynthesis and anaerobic or oxygenic photosynthesis and anoxygenic photosynthesis. So in oxygenic photosynthesis, like you can see the oxygen will be the byproduct. So same case. So when the student, like he added ferricyanate, there was an oxygen, right? So oxygen evolved. So here you can see the ferricyanate, it act as an electron acceptor and where it allowed the Hill's reaction there. Okay, so when we talk about the hydrogen acceptor, so here it is ferricyanate. Okay, so it got reduced and you can see water is there. So where water got oxidized to give us oxygen. Okay, so this is an Hill's reaction. And 129, final electron acceptor in respiration is. So very important question. So when we talk about the final electron acceptor in the respiration, it is oxygen. Okay, so it is oxygen. Substrate level phosphorylation occurs during which step of Krebs cycle? So when we talk about the Krebs cycle, in only one region, you can see there will be a substrate level phosphorylation. So where succinyl-CoA is getting converted to succinic acid. So that is in the presence of an enzyme, succinyl-CoA synthetase, right? So this is what we call it as substrate level phosphorylation, where GDP is getting formed there, right? So 131, fruit and leaf drop at early stages can be prevented by the application of, very easy one. So here the clue is, so early stage. So early fruit fall or early leaf fall, it can be prevented by applying auxin, right? Then next question. Which of the following acid is a derivative of carotenoid? So when we talk about the plant growth hormones, like we know 5 different plant hormones are there, right? In that, especially the one which is of carotenoid derivative is ABA, abscisic acid. 133. So assertion is vernalization is acceleration of subsequent flowering by low temperature treatment. Yes. Reason, the site of vernalization is apical meristem. So when we talk about this question, when we vernalization is artificial exposure, right? So when we subject the respective plant to low temperature, automatically you can see there will be a flowering. So which enhances to the seed production. And when we talk about the site where the actual vernalization takes place is meristematic regions. So shoot tip, root apex, like they'll come there. So reason is true, assertion is true, but it is not the correct explanation. Right? Reason is not the correct explanation. 134. The most abundant intracellular cation is. When we talk about this question, the most abundant intracellular cation will be K+. So 134, it is K+. 135, zygote develops a thick wall that is resistant to desiccation and damage and undergoes a period of rest before germination in. Like when we talk about the zygote, especially, like it goes with the desiccation, right? So you can see that especially in the algae and in fungi. So you won't observe this in a plant, gymnosperms or in angiosperms. Like you can observe this especially in the algae and in the fungi. Then 136 cleistogamous flowers are, when we talk about the cleistogamous flowers, like they won't open at all. So like when they are not opening, you can see anther and the stigma, like they'll be in close proximity. So when the flower is not opening, obviously like they have to go with self-pollination. So they are an examples of self-pollinated plants. 137, in a test cross involving F1 dihybrid flies, more parental type offsprings were produced than the recombinant type of offspring. So this indicates that. So we know when the genes are tightly linked, or when the genes are tightly linked and when they are present on the same chromosome, more parental characters will be observed and very few recombinant types will be observed there. So the two genes are linked and they are present on the same chromosome. XO type of a sex determination and XY type of sex determination are the examples of male heterogamety. So where male is responsible for producing two different types of gametes there. So one will have X chromosome and one will have Y chromosome. And 139, the X body of Henkin was observed in. So when we talk about the X body, like he observed this especially in the half of the sperms, right? Not in the full sperms of the insects. So he observed that X body in the half of the sperms, especially during spermatogenesis. So he actually observed that on the insects, right? 140. Given diagram represents the schematic structure of a transcription unit with some parts labeled as A, B, and C. So identify them. So when we talk about this question especially, you can see the one which is present on the A upstream region, it is promoter, and B is the template strand, C coding strand, and D will be terminator. Okay, which is on the downstream. Then the most abundant type of RNA in the cell is, so the most abundant type is ribosomal RNA. So you can see this is meant for catalyzing the protein synthesis, right? Nearly 80 to 85% of this RNA will be there. In total RNA, you can see 80 to 85% will be this rRNA. 142, which of the following statement is correct about the role of regulatory proteins in transcription in prokaryotes? So when we talk about the regulatory proteins especially, so they can act both as activators and repressors. So when we talk about the regulatory proteins, like they may positively regulate the process or they may negatively work also. So they may be an activator or repressor. 143. Consider the following statement and select the options. So when we talk about the first one, single cell spirulina can produce large quantities of food which is rich in protein, minerals, and vitamins. And body weight wise, the microorganisms Meteophilus, Meteotrophus may be able to produce several times more proteins than the cows per day. Yes, like when we talk about the correct one, first one is right. When we talk about the single cell protein, like where we'll be utilizing, they'll be rich in protein, minerals, and vitamins. And second statement is also right. Third statement, common button mushrooms are a very rich source of vitamin C. No, they are not of vitamin C. And a rice variety has been developed which is very rich in calcium. No, they are rich in iron content, right? So only two statements are right. 1 and 2 are right. One and 2. 144, in mung bean, resistance to yellow mosaic virus and powdery mildew were introduced by. So by which procedure, like we have introduced by via mutation breeding. 145, so we are supposed to match the bioactive substances and their role. When we talk about the statin especially, so this statin is used to lower the blood cholesterol. Right, so for A3 we got the answer. And cyclosporin, it is used as immunosuppressive agent. Streptokinase, it helps in removing the blood clot. Lipase, removal of oil stains. 146, given below are some examples associated with the type of adaptation. So we are supposed to identify which set of adaptation is this. So basking by desert lizards in sun, we know this. This is an behavioral adaptation. Even hiding in burrows by some animals, wearing of woolen cloth, thermal gapping, all these are the examples of behavioral adaptation. 147, when there are large number of post-reproductive or older individuals and less number of pre-reproductive individuals, then that the population is declining. When the number is less, especially for the pre-reproductive individuals, so that indicates you the population is declining. 148, a sedentary sea anemone gets attached to the shell lining of hermit crab. So this association is called commensalism. So when we talk about commensalism, like where one organism is getting benefited and the other is not getting benefited or it is not getting affected there. So it will be an commensalism. 149, rate of decomposition depends on. So rate of decomposition, it depends on all of this: chemical composition, temperature, soil moisture, and also the pH. 150, iodine is extracted from kelps, right? So from brown algae, laminaria, where this iodine is getting extracted. So with this, the botany part got over. Now like we'll start with zoology. Which of the following substance, if introduced into the bloodstream, would cause coagulation of blood at the site of its introduction? So you can see, very easy question. So prothrombin, fibrinogen, heparin, thromboplastin. So here, like when we talk about the heparin, so this is used as an anticoagulant. So it won't help in coagulation. And when we talk about the prothrombin and fibrinogen, all of them are actually in the inactive form, like they won't cause blood coagulation. It is thromboplastin. It is thromboplastin. Okay, so from the damaged cells, like you can see, we know the blood clotting procedure. So cascade reactions will take place there, right? So thromboplastin. 152. So the figure which is given here is adipose tissue. So when we talk about the adipose tissue, this is meant for storing the fat, and they'll also act as the insulator. 153. So from this question is very interesting question, like where we are supposed to identify the voluntary and involuntary muscles. So when we talk about the first one, when a boy jumping while playing, so that is voluntary. Pumping of the heart is involuntary. Making painting with hand, that is also voluntary, right? So 1 and 3. So they are voluntary, and involuntary will be 2 and 4. So movement of chocolate in your intestine. So it will comes under there in the involuntary action. 154, Brunner's glands are presented on the submucosa of duodenum. You can see this Brunner's gland will be there. Next question. Consider the following statement, each with, okay, for like filling the blanks. So bile duct and pancreatic duct, like they'll open into duodenum as common hepatopancreatic duct, right? So the one who is regulating is sphincter of Oddi, and DASH is a proteolytic enzyme found in gastric juice of infant. It is renin. And fatty acids, glycerol, we know like they are insoluble, so they have to get incorporated. So it is micelles, and again they'll get reformed into a small protein. We call it as chylomicrons, and where they'll be absorbed in the villi. So for this question, C will be the right one. So A, common hepatopancreatic duct, sphincter of Oddi, and micelles and chylomicrons will come there. 156. Dissociation of carbon dioxide from carbaminohemoglobin takes place when? So when the dissociation takes place, when partial pressure of carbon dioxide is less, right, and oxygen is high, obviously there will be a dissociation of carbaminohemoglobin. The respiratory center in the brain is stimulated by, so when we talk about the respiratory center, so it will be stimulated by CO2 concentration, especially in the arterial blood, not in venous blood. CO2 concentration in the arterial blood. 158, the chloride shift in red blood cells is the moment of. So when we talk about the chloride shift, or like we call this as Hamburger's phenomenon, right? So the chloride shift actually takes place when the HCO3, right, hydrogen carbonate ions, like when they diffuse out of the plasma, right? So to compensate that, like you can see the negatively charged chloride ions, like they'll shift it into the RBC, right? So chloride ions out of the cells to balance the release of hydrogen carbonate ions. 159. How many layers are present in the diffusion membrane of alveolus capillary? So when we talk about the total membrane, it is 3 there, right? We know thin squamous epithelium will be there, endothelium of alveolar capillaries will be there, and basement membrane will be there. 160. Which of the following events do not occur during joint diastole? So when we talk about the joint diastole, like you get first step will be all 4 chambers are, they'll be in a relaxed state, joint diastole. So you can see after that tricuspid and bicuspid wall, like they'll open. So where blood flows into the ventricles, then from the ventricles, they'll be poured into the respective region. So where semilunar valves will be closed there, and blood from pulmonary vein and vena cava flows into the left and right ventricles respectively through the left and right atria. So like when we talk about this question, all of these are right, right? All of these are right. So none of these. 161, hepatic portal system is. So when we talk about hepatic portal system, so this is a connection between the digestive tract and the liver. (163). Which of the following accessory excretory structures eliminates NaCl, lactic acid, and urea? So when we talk about this question, it is sweat gland. So who is responsible for eliminating the small quantity of urea and NaCl? 164. Juxtaglomerular apparatus is formed by cellular modification in the. So when we talk about this JGA apparatus, you can see this especially in the efferent arteriole and on the DCT, distal convoluted tubule. A fall in glomerular filtration rate activates. So when GFR, when the GFR is reduced, you can see the juxtaglomerular cells, they'll get activated. Okay, so again, JGA, previous question. So JGA cells, you can see them in the distal convoluted tubule and in the afferent arteriole. So if there is any fall in this GFR, it activates the JG cells to release the renin. Which of the following given ions are reabsorbed and secreted by DCT? So when we talk about the ions which are reabsorbed, sodium is reabsorbed and water is also reabsorbed and HCO3 is also reabsorbed. And H plus, K plus and ammonia, they are actually secreted. Okay, it is option A, which is the right one. So there will be a conditional reabsorption of sodium and water, like Dale takes place in the DCT, and also you can see there will be an a reabsorption of HCO3 also. (167) When a muscle bends one part upon the other, it is called. So like we call it as flexor. We call it as flexor. For an example, so when we talk about our hand, especially you can see forearm is there. So when we talk about the flexor movement, so this is responsible for bringing the forearm towards the upper arm. Find out the correct number of bones. Like in this question, like we are supposed to identify the number of bones. In the cranial region, you can see it will be 8. Facial bone will be 14 and hyoid bone will be 1. Middle ear 3. Malleus, incus and the stapes. So this weakness of muscles and bones in elderly occurs due to the deficiency of. So it is especially because of the deficiency of vitamin D. Next question. So you can see here a cricket player, he is chasing a ball. So which of the groups of bones will be directly contributing to this movement? You can see tarsals, femur, metatarsals and tibia. So he is chasing a ball. So the moment you can observe there and it will be there in the legs. So that will be the right one. Then 171. So we are supposed to arrange the muscle contraction events. So first one will be nerve impulse. Right? So nerve impulse will be generated. So once when the nerve impulse is getting generated, it automatically release the acetylcholine. Right? So that acetylcholine, it reaches the so nerve impulse. So where that acetylcholine, so it reaches to the synaptic cleft. So then from there, sarcolemma, which is having the receptor to send this acetylcholine, automatically the impulse it gets spread through the T tubules, and there will be a release of calcium from the sarcoplasmic reticulum, and the troponin, it will leaves the, and where it gets bind with the calcium, and there will be a contraction. And 172, corpus callosum connects. Like when we talk about the corpus callosum, like this connects the two cerebral hemispheres. Saltatory conduction of impulse occurs in, very easy question, myelinated nerve fibers. So in the myelinated nerve fibers, like you can see, there will be an conduction when, especially at the node of front wave. So it will be like a saltatory conduction. And when we talk about the Ivan Pavlov, like he performed the conditioned reflex. So he was a Russian physiologist, right? So he found the sight and smell of food reflex. So which made the hungry dogs to secrete saliva. So that is conditioned reflex. (175), you can see A, B, C, D, so parts of the eyes. So in the given figure, like you can see, A will be the retina, right? So this is the innermost layer which has photoreceptors. Rods, cones will be there. So that is right. And when we talk about the B, especially, B is blind spot. Okay, you can see in blind spot, there won't be any visual cells, right? So here they gave you as a few rods and cones will be there. Aqueous chamber, C, right? So when we talk about the aqueous humor, C is actually an aqueous humor. And when we talk about D, it is sclera, not choroid. Coiled portion of the labyrinth is called, so we call it as coglia. So the inner, when we talk about the inner ear, you can see it is coiled. So we call that portion as coglia. Next question. The blood calcium level is lowered by the deficiency of. So like when we talk about our calcium level, it is maintained by two important hormones, right? We are having hypercalcemic hormone and hypocalcemic hormone. So the blood calcium level is lowered by. So it is parathormone, right? So it is the deficiency of parathormone, like which leads to the lowering of blood calcium level. Next question. Gluconeogenesis, lipolysis, and proteolysis, like they are stimulated by. So they are stimulated by glucocorticoids. 179. Choose the correct statement from the following. Calcitonin regulates the metabolism of calcium. Yes, that is right. Oxytocin stimulates contraction of uterine muscles. Yes, that is also right. Graves' disease is caused by adrenal gland. No, it is by thyroid gland. Thymosin stimulates their absorption of water in renal tubules. No, this is done by vasopressin. Adrenaline, non-adrenaline, or emergency hormones. Yes. So in this question, the statement which is right are 3. 180. Hypothalamus axis. Assertion here is synthesis is oxytocin and vasopressin. Reason, neurohypophysis stores and release these two hormones. So when we talk about this question, especially, you can see the assertion is true. Okay. So when we talk about the oxytocin and vasopressin, especially, like that is synthesized actually there in the hypothalamus axis, right? And they are transported axonically to neurohypophysis. So here neurohypophysis is not storing that hormones. So from hypothalamus, like via the axonical way, where the hormones are getting transported. The seminal plasma is rich in. So seminal plasma is rich in fructose, calcium, and certain enzymes. 182. So we are supposed to identify the region which is responsible for producing sperm. So the region which is responsible for producing sperm will be testis, G. Right? Connects the epididymis with the urethra. So the one which connects the epididymis, you can see here, this is epididymis, right? Epididymis to urethra will be E. So that is vas deferens. And produces secretion that lubricates the penis will be from bulbourethral gland. Conducts sperms through the penis to the outside of the body will be the urinogenital duct, that is the external genitalia. Penis. 183. So this is from the menstrual cycle. So they have given you the representation of two hormones there. So we are supposed to identify those two hormones. So you can see, so the hormone after ovulation which is getting increased here is progesterone. So A must be estrogen there and B must be progesterone. In spermatogenesis, reduction division occurs during conversion of. So we know when we talk about the spermatogenesis process, it is the formation of spermatids, male gamete from the spermatogonium. So we know meiosis will take place there. Meiosis one is whom we call it as reduction division. So especially from the secondary spermatocyte, Yes. In this question, you can see, so in spermatogenesis, reduction division occurs during the conversion of. So when we talk about the reduction division, it occurs from the primary spermatocyte to secondary spermatocyte, like where it is getting reduced there, right? So which result in the formation of two haploid secondary spermatocyte. So reduction division occurs during the conversion of the primary spermatocyte to secondary spermatocyte. 185. Reproductive health in society can be improved by, so by introducing sex education, increased medical assistance, awareness about the contraception, equal opportunities to male and female child, encouraging myth and misconception. So in this question, you can see fifth one is wrong. So we are not supposed to encourage the myth, right? So remaining everything is right. 186. The permissible use of amniocentesis is for reduction of genetic abnormalities. So when we talk about amniocentesis, it is actually banned, right? It is permissible to detect the genetic abnormalities. And select the incorrect statement regarding in vitro fertilization. So when we talk about this question, especially ova from the donor male and sperms from the donor female and sperms from donor male are introduced to form zygote in the uterus, not in the uterus. Embryo with 2 blastomere is transferred into the uterus in fallopian tube. Embryo with more than 8 blastomere is transferred into the uterus. Yes. The baby thus produced is called as test tube baby. Yes. So 2 and 4, these two statements are right. The statements which are incorrect is 1 and 2. So in the question, we are supposed to identify the incorrect statement. Tasmanian wolf is a marsupial, while wolf is a placental mammal. So this shows. So when we talk about this question, especially, this is an important example which comes under the convergent evolution, right? So when we talk about the convergent evolution, like they may have different origin, but when we talk about the function, what they are performing, so it will be as per the area like where they got adapted. So it is convergent evolution. In the developmental history of mammalian heart, like we know, first thing is when we talk about the biogenetic law. So ontogeny repeats phylogeny. So ontogeny in the sense like the general structures like they'll get formed there. So this question is all about the same. So 2 chamfered fish, 3 chamfered fish, finally 4 chamfered stages there. So this indicates you the biogenetic law. 190. So this is from evolution. So when migration of a section of population to another place and the population occurs, dash change in the original as well as in the new population. So when an individual organism is getting migrated from one population to the other, the gene frequency, it gets changed there. So new alleles will be added to the new population, right? For new population, alleles will be added, and from the old population, like they'll be lost. So this would lead to the gene flow, right? So when it happens many times, like this leads to gene flow, and we call it as genetic drift. When it is taking place by chance, we call it as genetic drift. And when few organisms, like when they drifted from their original population and when they become founders, like we call it as founder effect. 191. SMAC is a drug which is obtained from. So SMAC is obtained from the latex of Papaver somnifera. 192. When an alien DNA is ligated in tetracycline resistant gene, the recombinant will. So insertional inactivation, right? So when an alien DNA is getting ligated into the tetracycline resistant gene, that alien DNA will lose the tetracycline resistant. During insertional inactivation, the presence of chromogenic substances will give blue colored colonies. So if the plasmid in the bacteria does not have an insert, so if they are not transformed You can see colony will be blue in color, right? So the blue color, it indicates you that is produced by the enzyme beta galactosidase. (194) Enzyme tag polymerase, which is used in PCR, has been isolated from the bacteria Thermus aquaticus. So if a species richness is indicated as yes, area A, Z is the slope of line. So the species richness will be shown as S equal to C A to the power, like you can see, Z will be there. Like when we take the log of this, so log S is equal to log Z plus Z log A. The region of biosphere reserves, which is legally protected where no human activity is allowed. We call it as core zone. And when we talk about the biodiversity hotspots, so you can see there will be high level of species richness, high degree of endemism, and accelerated habitat loss will be there. So the total number of biodiversity, that is hotspot, is 34, and in our country especially, you can see 3 hotspots are there, right? So when we talk about this question, both the assertion and reason are false. The UN Conference of Parties on Climate Change in the year 2011 was actually held in South Africa. Kyoto Protocol was endorsed at Conference of Parties 3. During the third session, like when we talk about the Kyoto Protocol, this is especially to reduce the emission of greenhouse gases, right? Then steps taken by the government of India to control the air pollution includes use of non-polluting compressed natural gas only as fuel by all buses and trucks. So with this, like we came to an end of the zoology part also. Thank you, everyone. Do well in your competitive exam. The exam will be easy. Do well. All the very best. And thank you very much.